Okay, we're going to get started because we're having that, I was just saying, we're having that post-lunch lag a little bit as people are trickling into the room. So we have two fantastic panels to finish up the conference this afternoon. And by the way, that patient panel was awesome, right? Yes, that was fantastic. And so if you liked that, I will also take the opportunity to plug Osteo, which is our AYA cancer podcast, which is like you're kind of just eavesdropping on these guys talking to each other, which is really fun. And also you'll learn a lot. So I'd love to introduce Sebastian Wang, who's a member of our MIB Agents Junior Advisory Board, and he will introduce our next moderator.
Good afternoon, everyone. My name is Sebastian. I'm 16 years old, and I'm the sibling of osteo angel Jaden. Jaden was diagnosed with osteosarcoma in his right proximal humerus, and he fought for 3.5 years, and he passed away last October. Today, I will be introducing Dr. Eugenie Kleinerman. Eugenie S. Kleinerman is a tenured professor in the Division of Pediatrics in the Department of Cancer Biology at MD Anderson Cancer Center. She holds the Mary V. and John A. Riley Distinguished Chair. She received an AB from Washington University in St. Louis and an MD from Duke University. Completed her pediatric residency at the Children's National Medical Center in Washington, D.C., followed by a fellowship at the NCI. She was a senior investigator at the NCI Frederick Cancer Research Facility, then recruited to MD Anderson. Dr. Kleinerman was head of pediatrics and Children's Cancer Hospital at MD Anderson from 2001 to 2015. Dr. Kleinerman is internationally recognized for her scientific and clinical expertise in osteosarcoma. She pioneered the use of MTP-PE, a unique immunotherapy agent for children with relapsed OS lung metastasis, resulting in the first improvement in survival in over 25 years. This led to MTP-PE's approval by the European Medicines Agency in 2009. Her research also focuses on understanding the mechanisms involved in osteosarcoma metastasis to the lung and how the tumor microenvironment inhibits immunotherapy. She is presently engaged in developing a dendritic cell vaccine for relapsed OS patients and the use of exercise to decrease the acute and late cardiotoxic effects of doxorubicin. She received the Distinguished Medical Alumni Awards from Duke University and Washington University, MD Anderson's Faculty Achievement Award in Clinical Research, the Sarcoma Foundation's Nobility in Science Award, and the Nolenberg Prize from SIOP. Welcome, Dr. Eugenie Kleinerman.
Thank you very much. This afternoon, we're going to get a glimpse into the future of immunotherapy, future targets, potential new targets, and future ways to stop the tumor's ability to block immune cells. Now, moderating this panel is not only an honor, but it's really rewarding for me, as it is a validation of my 40-plus year career in osteosarcoma and immunotherapy. So why do I say that? You are looking at someone who entered medical school wanting to be a general pediatrician. I emerged from medical school doing rheumatology, then allergy immunology, and now here I am focusing on osteosarcoma and immunotherapy. Lots of forks in the road, and I'm so glad I took all of them. Now, in the 1980s, when I was in the early part of my career, I was told that focusing on osteosarcoma would torpedo my career, that I needed to focus on breast cancer, lung cancer, colon cancer, because those cancers really mattered. And when I proposed that immunotherapy may be a way to get rid of the drug-resistant osteosarcoma cells, you can imagine the laughter in the room. I think it is really important to get some historical perspective. In the 1980s, if you could go to a study section and hear them talk about the ridiculous thought that immunotherapy could kill cancer cells and cure cancer patients, you would not really believe what you were hearing. So why am I telling you this? To make 3 points. First, I want to talk to all you young investigators and physicians. If you see a fork in the road that really resonates with you, listen to your heart and take it. Number 2, if they tell you you are wrong and you are crazy, but you really, really believe in what you're doing, do it. Be stubborn. Be persistent. But there's a caveat to that. Don't expect it will be easy. Don't expect that you'll be in the in crowd, and it may be a lonely path. But I promise you, when you emerge, in the words of Shirley Temple, you will say, "Oh my goodness, thank goodness I took those forks." My third point is to MIB agents. Thank you for the funding you provide to give young investigators, some of whom are on the podium here, hope and support. Thank you for allowing them to pursue their perhaps sometimes crazy ideas. Thank you for educating the world that osteosarcoma matters and for elevating the voices of patients and parents. Now back to the future of immunotherapy. We're going to first hear from Dr. Richa Kapoor, a scientist at Children's Hospital of Philadelphia, on how defining the immunopeptidome of osteosarcoma can identify new therapeutic targets. Dr. Geoffrey Smith from Boston Children's Hospital is going to talk about the evasion of CD8 positive T-cell responses in murine osteosarcoma. Dr. Betsy Young, who was one of last year's Young Investigator awardees from the UCSF Benioff Children's Hospital, is going to talk about how cGAS-STING pathway drives immunovation in osteosarcoma, and to discuss how this pathway may be a therapeutic target. Dr. Brian Mooney, a research associate at the BC Cancer Research Center, will talk about another new potential target, the receptor tyrosine kinase-like orphan receptor 2, and how this may be used in immunotherapy. And finally, we're going to end with Dr. Arianexis Aquino-Lopez from the Baylor College of Medicine. Her talk is going to be on repurposing virus-specific T-cells as a novel immune therapy for osteosarcoma. Dr. Kapoor?
Hello, everyone. First of all, I would like to thank the Factor 2025 organizers for giving me this opportunity to present my research aimed at defining the immunopeptidome of osteosarcoma to nominate candidate therapeutic targets. So just to give a brief overview of immunotherapy, immunotherapies which are in development for osteosarcoma is targeted against a number of cell surface molecules which are commonly overexpressed on these cells, and accordingly a number of antibody-based or cell-based immunotherapy approaches are being tested for this disease. However, currently no effective immunotherapy is available because of a probable lack of optimal immunotherapy targets. So here at Children's Hospital of Philadelphia, in the Maris lab, we recently discovered a novel immunotherapy approach for neuroblastoma, a similar solid cancer, and I would like to give you a brief overview of that immunotherapy. So here, a CAR T-cell based therapy has been developed where a 10LH scFv, a CAR, targets a 9-amino acid peptide derived from FOX2b, and it is presented on 21 different HLA alleles. And here in the green, you can see the scFv targeting the peptide in red, presented on HLA A2402. So the IND for this peptide-centric CAR T-cell therapy was approved in May, and we will be dosing our first patient in August. Just to give you a brief overview of how the preclinical data looks like for this CAR T therapy approach in neuroblastoma, here on the left you can see tumor growth curves, and on the right, you can see the T-cell count in the blood monitored over time. These FOX2B peptide-centric CAR T-cells showed complete sustained regression of highly aggressive neuroblastoma models. Here indicated in blue are the mice which were treated with the CAR T-cell therapy. And here in the COG model, you can see one of the mouse relapsed, but was subsequently treated with a second dose of CAR T, and we monitored these mice for 120 days and saw complete sustained regression. Having this effective immunotherapy in our mind, we wanted to extend this approach to osteosarcoma and wanted to first check the HLA expression of osteosarcoma models as compared to neuroblastoma. Very interestingly, at the RNA level, as well as at the protein level, we saw high expression of HLA indicated in the yellow and red in the osteosarcoma as compared to neuroblastoma in the blue. And this perfectly correlated well to the protein expression. Hence, with the hypothesis in mind that the osteosarcoma immunopeptidome consists of peptides derived from proteins critical to the bone development, which are either unique to this disease or are upregulated due to aberrant epigenomic alterations characteristic of this disease, we have this rationale that the tumor-specific peptide HLA complexes are targetable with peptide-centric CARs, and hence we wanted to perform deep proteogenomic profiling. And hence, in collaboration with Dr. Alejandro Sweet-Cordero's group at UCSF, we performed immunopeptidomics on 16 osteosarcoma PDXs in 3 cell lines, and these PDXs belonged to different HLA-A allotypes, which are commonly occurring in the patient population. And then, having identified unique peptides from different osteosarcoma PDX models, we narrowed down on 20 different peptides based on a criteria of statistically significant peptides, and those peptides should be presented by more than 2 PDX samples, and the parent gene presenting that peptide should be high in osteosarcoma, and that peptide should be absent in the normal ligandome. So I would like to give you a brief overview of the targets which we have identified so far. So we identified several different peptides derived from proteins PANX3, SP7, TLX3, and LRRC15. Here towards the bottom, in the red, you can see a peptide derived from LRRC15 to be presented in several different PDX samples. So just to give you a more clear picture of this complex heat map, I would like to mention on the very left, we have the PDX samples presented the peptide. And next comes in purple is the TEPAP score, which indicates the high importance of that specific protein in osteosarcoma. The peptide derived from LRRC15 was found to be absent in the immune epitope database, meaning in the normal tissues, we did not see the presentation of that peptide. And while on the right-hand side, we see in red the RNA expression. It was very high for LRRC15. And what makes this peptide-centric CAR T therapy approach very novel is that, however, there is a dirty RNA expression of LRRC15 in some of the normal tissues, but there was no peptide presentation by the normal tissue in this case for that specific peptide. And that peptide was interestingly known to be and seen to be presented by several different HLA-B alleles, which are of common occurrence. And next, we wanted to see the expression of LRRC15 at the protein level, and we performed immunohistochemistry, and that indicated high level of this protein in osteosarcoma, and we also identified this protein in other sarcomas like rhabdomyosarcoma and Ewing sarcoma as well. Next, we wanted to validate the peptide HLA complex indicating strong binding of this specific peptide HLA using different peptide epitope validation assays, and we are also able to refold this peptide HLA complex using size exclusion chromatography, and we were able to isolate huge mg of protein. While also we validated the thermal stability of this peptide HLA complex, which was pretty stable. And hence, moving forward with this approach, we also looked at several other targets like SP7, which we found is an osteogenic driver in osteosarcoma. It's an oncogene, and we also identified huge expression of this protein in osteosarcoma, and a peptide derived from this protein is presented by a highly recurrent HLA-A01, and we validated that peptide MHC complex. While interestingly, some peptides identified in neuroblastoma were also found in the osteosarcoma immunopeptidome. So it was interesting that we have the peptide-centric CARs for these peptides derived from protein PRAME and IGF-people1 already in the lab, and then we performed killing assays, and we saw here with IGF-people1 CAR specific killing of the osteosarcoma cells, while no off-target killing. While similar approach for PRAME CAR T cells, we observed specific killing at different effector-to-target ratios in osteosarcoma cells, which specifically present the peptide like OS525 and U2S, and there was no off-target killing in the osteosarcoma PDX-derived cell line, which did not present this peptide. Having all this in hand, we wanted to know and explore how much of the osteosarcoma patient population we will be targeting by this peptide-centric CAR T therapy approaches in hand. So around 56% of the osteosarcoma patient population in North America, we will be able to target with the present peptides, which we have identified till now, and we aim to extend it to 100% of the population in near future. Hence, our study has identified numerous peptide targets which can lead to the generation of potentially impactful new cellular immunotherapies for osteosarcoma. And currently, we are performing whole exome sequencing, RNA sequencing, and immunopeptidomics on human osteosarcoma primary patient samples. We are also identifying public neoantigens by looking at the non-canonical peptides, while there is an ongoing discovery effort in collaboration with Mediotherapeutics for the validated peptide HLA complex for LRRC15. While interestingly, the SP7 peptide harbors a cysteine, which makes it sticky and challenging for performing phage display. So we are trying to find out ways differently we can approach to have a binder for the CAR T cell therapy targeting SP7-derived peptide. I would like to thank the Maris Laboratory and Dr. Alejandro Sweet-Cordero's group at UCSF for providing me this guidance and support to do this research, while also thank Mediotherapeutics, the CHOP Proteomics Core, Dr. Yael Muzay, and Dr. Nick Sarakis at CHOP, and Dr. Mark Yarmarkovich at NYU, and also my funding agencies. Thank you.
Thank you. First, thank you to Christina and the organizers for a chance to share my work today about the evasion of endogenous CD8 T-cell responses in a murine model of osteosarcoma. And then a particular thanks to Dr. Kleinerman's introduction, because it resonates, since I've been told many times that there will be no endogenous responses in this model, and you cannot see a T-cell response to osteosarcoma. I hope I will convince you otherwise. So why is this interesting? There's been a revolution in many, mainly adult cancers, using immune checkpoint blockade agents to unleash a patient's immune system to attack their cancer and provide long-term control. We've seen changes in the standard of care in non-small cell lung cancer, in melanoma, in many genitourinary cancers. These work by blocking molecules either on the T cell, like PD-1 or CTLA-4, or by tumor or APC ligands like PD-L1 with monoclonal antibodies. Unfortunately, osteosarcoma has not benefited from this. There have been trials of pembrolizumab, and across two of them, there have been only 2 published partial responses, which you can see in the waterfall plot on the far right. There have also been trials of the other PD-1 agent, nivolumab, with no responses, and trials of CTLA-4, also with no responses. So when I finished up my clinical year in fellowship, I was interested in the broader question of why is this? And to address that, I joined Tyler Jax's lab at MIT, who has pioneered using autochthonous mouse models to study the relationship between a developing tumor and the immune system. And it's important, obviously, to have an immunocompetent model system to be able to address this question. So my colleague, Matthew Epinet, introduced this model to you yesterday, and it was originally described by Jackie Lee's lab and Stu Orkin's lab concurrently in 2008. But briefly, it's P53 and RB loss under control of the SP7 or Osterix-Cree. This yields in our hands a relatively human-like distribution of osteosarcoma, with about half the tumors occurring in the hind limb. The histology is also predominantly osteoblastic and expresses the tumor marker SATB2. And we see about 20 to 30% of these mice have pulmonary metastases that calcify and can be detected on micro-CT. As Matthew described yesterday, we've now characterized the genomics of these tumors and see that they reproduce some key features which will help, I think, address some of the critics and skeptics about whether you can generate a response. That is to say, there is a low tumor mutational burden in this model and yet a relatively high structural variant rate, comparable to the human disease. We've also been able to derive several cell lines, as other groups have done as well, which allow you to do more kind of scaled preclinical studies more rapidly than this model that is otherwise notoriously slow. So the first question we asked that was very important was, what role does adaptive immunity play in developing osteosarcomas? And so we crossed this model to the RAG2 deficient mouse that lacks B and T cells and so has no adaptive immunity, but has intact innate immunity. And what we see is a consistent, small, but about 30-day delay in the onset of osteosarcomagenesis. And this is regardless of what genotype of the mouse you use. I'm showing here the heterozygous for RB, so there's a slightly slower model than some of them, but it is consistent across all of them that it's about a month, suggesting there's an initial period of recognition by the immune system where it controls the tumor, or maybe it eliminates some very sensitive clones, but then eventually all these mice succumb to the tumor. We can also do this in the transplant setting, so we can take a slow-growing line, which was our 254 line. This is a line that when you put it in an immunocompetent mouse, it actually barely grows out. You can see it by micro-CT, but the animal ultimately is able to control it. But by contrast, if you put that same cell line in a RAG2 mouse, you see basically uncontrolled growth. If you take a more aggressive line, like our 383 line, it grows out in the immunocompetent setting as well, but it grows out sooner and the tumors are much larger in an immunodeficient mouse. If we look at those mice that control tumors, that 254 tumor, we can see that there's actually development of immunological memory. So one way to do this is to look for cytokine production by the T cells that were in that mouse after you challenge them with that same cell line again. And so we did ELISpot assays. And what you can see is when you re-challenge those T cells from a mouse that had rejected the tumor, they produce a strong interferon gamma response. What is also interesting and potentially therapeutically relevant is even when you challenge them with the related cell line that they've never seen, that 383 cell line, they do generate some degree of an interferon response, suggesting there may be some common epitopes or misexpressed proteins that the immune system could be recognizing. So then why, despite this initial period of control and this evidence of memory formation, do 100% of the immunocompetent mice ultimately end up with large tumors? To begin to address that, we've been looking in the late-stage tumors, initially by flow cytometry, and so we were able to make single-cell suspensions and figure out what is the immune composition of these tumors. At first pass, if you look at the tumor infiltrate, there are CD4 and CD8 T cells here. They're not a lot. They're about 10,000 per tumor, but they are definitely there. We can go on to start looking at the phenotype of these T cells. And so you can look at phenotype in a few ways. One is you can look for expression of effector molecules like granzyme B, which a CD8 T cell will use to kill cancer cells. And what we saw early on was actually this is pretty strongly upregulated in the tumor-infiltrating lymphocytes that we pull out, with something between 20 and up to even 80% of the CD8 T cells expressing granzyme B. And that's much higher than you will see in CD8 T cells you take out of a control lymph node or of an unaffected bone. We can also look at exhaustion or activation markers and see evidence that these cells are likely encountering antigen in the tumor microenvironment. So they are more Ki-67 positive, indicating that they have seen antigen and are likely proliferating. They also have upregulated PD-1, which is one of the first things you see after activating the CD8 T cell. And then you can see on the plot on the left also they're all CD44 high, which is another canonical marker of antigen experience. So we seem to have some in these tumors when we grind them up. They seem to be activated. So maybe they're in the wrong spot. So the next thing we asked was, well, where are these tumors relative to the cancer cells? And we did multiplexed immunofluorescence with Sandro Santagata's group at Brigham, and what you can see here are the SATB2 positive cancer cells are co-localizing with your green CD8 T cells that also express granzyme B in yellow. So they seem to be in the right place. So we have cells there, they're encountering antigen, they're making the effector molecule, and they're next to the cancer cells, yet the cancer cells are not dying. So what's going on? I don't have the final answer to that, but I'm going to give you a couple of hypotheses that we're following up on. The first one, I would say, is an idea of poor T cell priming in these tumors. So we've conducted single-cell RNA sequencing and single-cell TCR sequencing on the T cells from these late-stage tumors. And what you can see is predominantly in this population 0, they have what we call a naive or early activation-like phenotype. So they no longer express true naive markers. They're not TCF7/TCF1 high, but they haven't become exhausted. They're not expressing tox. These are the same cells that are expressing that granzyme population. And so they're kind of stuck in this early state where they've never been pushed into a fully activated response or an exhausted response, which is normally what will give response to immune checkpoint blockade. When we look at the T-cell receptor distribution, and we can see what we call an oligoclonal response. So if you have a strong single antigen expressed in a tumor, you would expect the majority of the T-cells to be from 1 or 2 clones that are recognizing that antigen. We see that in only really 1 mouse of the ones that we sequenced, and they're all in that one little island of a memory-like population. The rest of them in that cluster 0 are all relatively oligoclonal, so you do have some expansion, but not a lot, suggesting that there hasn't been strong priming of a single response. So I think this is a big part of the problem, and I think we're hopefully going to follow up on some of the antigen identification work that Dr. Kapoor mentioned and do it in this model and understand what are they recognizing. So I hope there's more to come on that. The other area, of course, is the tumor microenvironment, which we've heard a lot about at this meeting, and it's now very obvious that the tumor microenvironment is not just osteosarcoma cells, it's the T cells we've been talking about, it's B cells, it's macrophages, it's neutrophils. If we look first at some of the other T cells, we do see an increase in regulatory T cells, which are an immunosuppressive population, usually meant to prevent autoimmunity, but we know that they can also protect cancer cells from being eliminated. And so compared to healthy control settings, there are more FOXP3 positive Tregs in these tumors, and there are also more from this particularly immunosuppressive KLRG1 positive population that our lab has shown in non-small cell lung cancer actually prevents a strong CD8 response. So this is potentially another part of the problem. If we look over to the myeloid compartment, which others have talked about, this is also likely where some of the problem is, at least in these late-stage tumors. So we can see that about half of these cells are neutrophils, which we have not looked closely at yet. But the other part are a lot of macrophages, which you could also see on that multiplexed immunofluorescence I showed earlier. And if we dig into the phenotype of these macrophages in a simplistic way, we can see that most of them have more of an M2-like phenotype. That is to say that regardless of whether they were tissue resident or embryonic in origin, which are the majority of the cells in there, or this tissue resident, they express high levels of arginase 1, high levels of CD206, and high MHC2, which all mark a more immunosuppressive and less anti-tumor population, and they express low levels of INOS and CD86, which usually help control a tumor. So with that, I want to close and just say figuring out the answers to this, I think, are important because they suggest barriers to potential therapies, and which you're hearing more about today. So if we think about the first model that there's poor priming, you could consider vaccinating against some of those antigens and getting a bigger endogenous response in the patient. Or you could think about developing that response ex vivo with a CAR T-cell or an adoptive cell therapy strategy. For regulatory T cells, there's now a clinical agent to potentially deplete these from tumor-infiltrating settings with a CCR8 depleter. And macrophages can be eliminated through things like CSF1R antibody depletion, or as you'll hear later, repolarized with things like STING agonists. So with that, we'll give a big thanks to Tyler Jacks for letting me start this project in the lab and drag him into the osteosarcoma world, and a huge thanks to Matthew Epinet, who's done a lot of the beautiful work you saw yesterday and today, and thank you all for your attention.
Hi, everyone. Good afternoon. I'm so happy to be here at Factor again this year. Thank you so much to Christina and all of MIB for the opportunity to share some, I think, exciting interval updates on my work studying STING and osteosarcoma. I'm a physician scientist and pediatric oncologist at UCSF and a postdoc in the Sweet Cordero lab. And I've actually been studying cGAS-STING and osteosarcoma for some years now. And today, I pushed myself to really just show totally new data, and I'm really going to be focusing on one specific part of our story, which is really studying STING agonism in murine immunocompetent models, and some of the immune characterization that we've done therein. Also want to just say a quick thank you to the Family Fund because of Charlotte. It's been an honor to perform this work with your support over the last year. So the cGAS-STING pathway is present in tumor cells and normal cells, and it can be repressed or activated to regulate anti-tumor immunity. We originally developed an interest in studying the cGAS-STING pathway because of the link between chromosomal instability and cGAS-STING activation or repression. And really this canonical pathway becomes activated either in a tumor-intrinsic manner by double-stranded DNA in the cytosol, or in the context of therapy such as STING agonism. When STING is activated, basically a type 1 interferon response occurs, and this can really activate anti-tumor functions in a number of immune compartments, including myeloid cells as well as T cells. So the overarching goal of this work has really been to define the function of cGAS-STING in osteosarcoma in a couple of different contexts, including tumor evolution and response to therapy. And I think we've made great strides in all of this, but I wanted to take a moment for my talk today really just to focus on the bottom corner in green, our work looking at the role of STING in the response to STING agonism. So the models that we employed here are likely well known to this crowd. We used 2 different murine immunocompetent cell lines in our orthotopic model, K7M2 in bulb C and F420 from the Eugenie group in black 6. And I think this is really important to our conversation yesterday about rigorous experimental models to really use at a minimum 2 different models for immunology-related studies to really try to recapitulate different sort of immune skews that are present in the mouse models that we have at our disposal. And we used the STING agonist called Diabzi, which is an IV STING agonist, which is quite unique from intratumoral STING agonists that have been studied for quite some time. So first, when we treated K7M2 in bulb C mice with orthotopic hind limb tumors with 1 dose of Diabzi, the STING agonist, we actually saw a significant control of tumor growth with just this very sort of limited therapeutic approach. So then we moved forward to employ an isogenic cell line pair that we had generated with tumor STING knockout. And in this case, we treated the tumor-bearing animals with 2 doses of Diabzi separated by 1 week, and we actually saw long-term survival following complete tumor regression in this model. And I think to our surprise, we actually found that tumor STING was entirely dispensable for this effect. So in this survival curve, you can see that a majority of animals experienced long-term survival when treated with the STING agonist with or without the presence of tumor STING. So this really suggested to us the importance of the host STING response in the tumor microenvironment, perhaps, or systemically, given that this is an IV STING agonist. So we moved forward to try to characterize some of the first responses that we saw after treating these animals with the STING agonist. So we performed some profiling of the tumor microenvironment actually at a very early time point, 2 hours after Diabzi, as well as 48 hours. And we observed across these 2 time points, as well as others, that there was an induction of monocyte recruitment to the tumor microenvironment, and kind of a commensurate decrease in neutrophils, as well as TAMs that we saw both at the 22-hour time point and then later at 48 hours as well. It's actually been shown in literature in other cancer contexts that DCs and CD8 T-cells can be acutely depleted in response to STING agonist, and we observed that as well. But interestingly, we saw a decrease in Tregs as well at the 48-hour time point, and I'll come back to Tregs in a moment here. So I think this was an early indicator to us that given that the tumor STING really didn't seem to be the major player in the responses we were seeing, that perhaps the monocyte induction due to monocyte STING may be responsible. So then we moved to a number of sort of perturbation studies looking at the role of CD4 and CD8 T-cells in the efficacy of Diabzi, because we were wondering, even though we saw this monocyte recruitment, given that we experienced this complete tumor regression and long-term survival, perhaps adaptive immunity was also implicated. So initially, we treated the same tumor model with an anti-CD8 with and without Diabzi, and I think this gets to some of the content from Jeff's presentation also, that when you deplete CD8, you do get quite an aggressive growth phenotype. However, treating with Diabzi was actually able to significantly improve survival, even in the absence of CD8, and really was able to sort of move the needle on this model that was even more aggressive. So then we moved to CD4 and found quite a surprising result. When we treat with the CD4 depletion, we saw really the opposite entirely, that it seems that CD4 is actually antagonistic to the efficacy of Diabzi. So the mice that had a combination of Diabzi and CD4 depletion experienced basically the best response of any group in any of these models. So suggesting here, perhaps through a mechanism involving Tregs suppressing other immune cells or exerting effects through immunosuppressive cytokines. And then just to round out sort of the list of experiments for completeness sake, we also wanted to do a combination experiment of CD4 and CD8 depletion together. And this gets tricky to think through, I think, since what I've shown on the prior slide is that there's really kind of completely different effects of 4 and 8 separately. But basically in this study, we really were left with kind of an intermediate result that didn't parse out in any specific way, which I think in follow-up to the prior findings made some good sense. So then we moved to the concept of immunologic memory, which you've also heard some about, and what we had identified with our initial studies in which we administered Diabzi twice and then experienced this long-term survival phenotype, that when we re-implanted the same cell line, 100% of mice rejected these tumors, and that's shown on the left plot. So then moving to the right plot, when we repeated this again, you can see the growth curve of the naive animals that have not previously seen this tumor. And then when we depleted CD8, we surprisingly also observed that 100% of the animals still rejected this tumor reimplantation. So this was quite surprising to us, that it seemed like in our hands in this model with Diabzi, the long-term immunologic memory that was conferred by this STING agonist did not seem to be CD8 T-cell dependent. So then we moved forward to test this again with a combination of anti-CD4 and anti-CD8 in this re-challenge setting. And we observed now that basically the mice kind of universally succumbed to those tumors in a similar manner to the naive animals, and in quite a different manner than the re-challenge mice that did not have depletion of these cells. So the key portion to this that's still pending is the CD4 alone in this re-challenge setting, and I think with sort of the combination of all of these experiments, we'll have the most sophisticated window into which of these cell types are contributing to the adaptive immunity that we do believe in some way is kind of interacting with the innate immune mechanism of Diabzi. And then lastly, because I shared that using different models is quite important, we definitely wanted to test this in the F420 Black 6 model. This is quite an aggressive model, and we were able to see that with 2 doses of Diabzi, we did see some limitations in the growth and a significant survival benefit, but we're quite interested in pursuing this in follow-up, perhaps starting the STING agonist earlier after engraftment, and also studying this in the metastatic setting. So in summary, this is the model that we've proposed to date, where Diabzi exerts this powerful anti-tumor efficacy in murine models of osteosarcoma, characterized by kind of an initial monocyte-driven innate immune response, followed by an adaptive response that may in some way contribute to immunologic memory, and a model wherein CD4 cells are likely to actually be playing a pro-tumor role and are antagonizing the effects of Diabzi in the acute setting. And so this is just another summary of the work to date, and we have a manuscript that is in the end stages of the revision process, so we're super excited about that. Also thinking a lot about how these sort of immune perturbation studies can really allow us to inform ongoing clinical trials on STING agonism for osteosarcoma, and also thinking that because there are acute toxicities with this sort of innate immune activating therapy, how can we work towards combination therapies to be able to de-escalate the dose in certain scenarios. And thank you so much again to the Family Fund Because of Charlotte, as well as those folks from Alejandro's lab group who have been really instrumental in my development as a physician scientist. Just a quick shout out to Christine, my incredible technician of 3.5 years, who's starting at Scripps grad school in a couple of weeks here. And also because Troy's in the audience, shout out to him as well. And thank you all so much.
Okay, good afternoon, everyone. So first and foremost, I just wanted to thank the organizers for inviting me back. I was fortunate enough to attend this meeting last year, so I feel really happy to be able to be invited back. It's such a wonderful conference. So today I'll be giving a bit of an update on the proteomic work that our lab has been doing to identify new immunotherapeutic targets in osteosarcoma. So a lot of this work, kind of the genesis of my project came about from this study here. So this publication used genomics and transcriptomics to characterize about 260 models of pediatric cancers. And we learned a lot about pediatric cancers with this study. However, what we wanted to do with these models was utilize proteomics to try to identify the surface candidates and see if any of those would be exploitable for specific different types of cancer. And the way the cohort looked was a little bit like this. So the Children's Hospital of Philadelphia processed all of these samples on the left-hand side, and then the team at BC Cancer processed all of these samples on the right-hand side. So if anyone in the audience is interested in any of the other cancers, do come talk to me afterwards. But today I'll just be talking about the 22 patient-derived xenograft models of osteosarcoma that we processed. In terms of how we process them, it's not overly important to know how all of this works, but I did just want to show the actual methodology. So we use a density gradient ultracentrifugation approach. So we enrich plasma membranes from the PDX models of disease, and then ultimately use mass spectrometry to identify what those proteins are. Now, what is worth knowing is the control that we use. So we do use a reference mixture of about 12 non-osteosarcoma cell lines. We prepare this identically to our osteosarcoma PDXs, and we process them as seen here. So a lot of the statistical analysis that you'll see in the coming slides will be compared to this control here. In the interest of time, I'm going to switch over this and just go straight into the identification of the targets. So the first question that we had was, do we identify known candidates in osteosarcoma? And the question is yes. So I'm sure many of you in the audience will recognize many of these targets like MMP14, CADM1, and CD276 or B7H3. But really, we want to ask the question, how can you utilize this data to try to identify the lesser studied or even new surface candidates? And typically what you would do in this situation is generate these lovely volcano plots, which show the proteins that are enriched in your PDX versus your control. And this is a really good visual aid to find what's there and what's not. However, it doesn't really get to the crux of the issue that we're trying to address. The first one being, we don't know how many of these surface candidates actually have targetable real estate on the outside of the plasma membrane. And the second, and perhaps most important, is we don't know the normal tissue expression of these. So one of the tasks I had during my postdoc was to try to figure out some sort of a scoring system to prioritize these candidates. And I did actually show the scoring system last year in Factor, but I think 1 minute to try to describe a scoring system just doesn't really work. So if anyone has any particular questions about how I did it, do come find me afterwards. But all we need to know here is the candidates on the left-hand side of this plot here are the candidates that are really highly enriched in osteosarcoma, but also low in normal tissues based on the GTEx data. It's the data we have available to us. And then the candidates on the right-hand side would likely still be enriched in osteosarcoma, but are probably high in normal tissues. So when we do this, we identified the second highest hit in our data was this candidate known as ROR2. ROR2, it's a bit of a mouthful. It's the receptor tyrosine kinase-like orphan receptor 2. It's a 130 kilodalton pseudokinase, and its predominant role is in non-canonical Wnt signaling. So it seems to be involved in relaying the signal from particularly Wnt5a, and this is involved in limb development prenatal. It does have an important paralog, which is ROR1. Those of you might know ROR1. It is under preclinical investigation as an antibody-drug conjugate and CAR-T in some adult cancers. The structure between ROR2 and ROR1 is very similar. In terms of ROR2's role in cancer, the overwhelming majority of the data would suggest ROR2 plays an oncogenic role. There are a handful of sort of smaller studies suggesting a tumor-suppressing role. But from what we can see, there's a few limited studies in osteosarcoma, and they all suggest ROR2 plays a role in the oncogenic phenotype of osteosarcoma, though these studies are limited. And one of the things worth pointing out here is there is a phase 2 clinical trial, which I believe just finished, that's being run by BioAlta. They're trying to develop an agent called ozuriftamab vedotin. It's an MMAE conjugated antibody, and they are testing this in head and neck squamous cell carcinoma adult patients. So that was quite interesting for us to see, but we wanted to see if we could sort of work ROR2 out in the pediatric setting. So in terms of the data that we have available to us, we do see high expression of ROR2 in osteosarcoma versus other pediatric cancers. We do have cell lines that readily express ROR2 in the lab, and we can knock ROR2 down using siRNA. When we knock ROR2 down, we do see a nice reduction in the proliferation of cells, both in the short-term incucyte assays and also the longer-term clonogenic assays. We have also expanded this to 3D spheroid assays. I'm not showing the data here today, but we do see the same phenotypes, which is really nice to see. And we also see that the cells do seem to migrate less when we knock ROR2 down, though this isn't statistically significant in one of our cell lines. We did expand to the ex vivo pulmonary metastases assay, which Dr. Lizardo gave a really nice presentation on this morning for those of you that were in attendance. But this allows us to look at the ability for cells to propagate the mouse lung microenvironment. And so we generated these shRNA cell lines, and we looked at the ability for them to propagate. And what we were really nicely be able to show was loss of ROR2 significantly impacted the ability for the cells to propagate the lung microenvironment in these PUMA assays. So up to this point, we were pretty happy with the data we were seeing. We were showing that it does seem to have an oncogenic role in osteosarcoma, and knocking it down does seem to inhibit the growth and the migration somewhat in cell line models. But what does its expression look like in normal tissues? So we did investigate that in about 25 normal pediatric tissues, and we are really happy to see that really limited expression of ROR2 in the normal pediatric tissues when compared to the osteosarcoma samples that we had available to us. And that is the data quantified there for those of you that are interested. So at this point, we think we have a pretty interesting target. So it does seem to play an oncogenic role, and it seems to be really not expressed in normal pediatric tissues. So where do we go from here? And so what we decided to do was try to identify novel binders of ROR2 using phage display biopanning. I'm very much going to try cram over a year's worth of work into 2 slides here, so I will just have to verbally explain some of it. But we used phage display biopanning in collaboration with our wonderful collaborators at UPIT to identify novel binders of ROR2. And what we were able to find was a number of really successful agents that were capable of recognizing ROR2. The 2 that we decided to take forward for further investigation was the FAB44 and FAB50, which you can see on the right-hand side of the graphs. The reason we took these forward is because we saw preferential binding of these to the osteosarcoma cells expressing ROR2, but not the cells that weren't expressing ROR2. I'm just not showing that data here today, but that was some flow cytometry data that was supplied to us by our collaborators in UPIT. What we did was we created the humanized antibody version. So we created the IgG version. So these were IgG44 and IgG50, and then used these internalization assays to confirm that these are indeed internalized into OS742 cells. And we see that the internalization does seem to be diminished when we knock ROR2 down, suggesting that it is dependent on the expression of ROR2, which was quite nice to see. And then the final thing, which, excuse me, I could probably spend 10 minutes talking about this alone, but one of the reasons we're interested in developing our own binders is the idea of trying to utilize new payloads rather than just the MMAE payload. So this is just data from one cell line, but we've expanded this to multiple different cell lines where we've been looking at whether there's a differential response to common payloads that are currently being used. And what we were able to see was a really nice response to payloads such as the PNU 159682, which is a nemorubicin derivative, and also the really strong PBD. So with that, I would just like to conclude that I believe the surfaceome profiling done a very good job of identifying both known and new surface proteins in osteosarcoma. We believe ROR2 is enriched in models of the disease, and we do think it plays a role in both the proliferation and the migration of osteosarcoma cells. And we've now turned 2 of our ROR2 binders into antibody-drug conjugates. So we're bang smack in the middle of our in vitro and ex vivo assays, trying to assess the efficacy of these prior to going in vivo. But hopefully, if I get an invite back to next year's Factor, I'll be able to give an update on that. And so with that, I would just like to thank everyone that was involved in the study, particularly Paul Sorensen and the Paul Sorensen lab. It's a really wonderful lab to work in on things like this. The proteomics lab, Dr. Greg Moran, and our wonderful collaborators at UPIT, Wei Li and Xiao-Chi Chu. And finally, I would just like to thank the MIB Family Factor Travel Award, which supported my travel here. So I was here because of Edward. So thank you so much, and I look forward to discussions later on. Bang on time.
Good afternoon, everybody. Thanks, MIB, for the opportunity to present our work here today. And thanks to the Kaplan family. This work is because of Sydney. And I'm excited to share a little bit about my project in where I'm looking into repurposing virus-specific T-cells as an immune therapy for osteosarcoma. So this audience doesn't need a lot of introduction and the special need of developing new therapies for osteosarcoma. We know that one of the biggest challenges is once our patients develop metastatic disease to the lungs, right? And so we know that our overall survival has been at the 60% for decades, and with even worse survival, less than 40% once our patients develop metastases. So because of that, I'm particularly interested in developing novel immunotherapies, in particular, virus-specific T-cells. And so why virus-specific T-cells? There's been work showing that virus-specific T-cells are resident cells in a variety of solid tumors. So these cells are already living at the tumor site. This has been shown in multiple tumors, endometrial cancer, head and neck, glioblastoma, breast cancer, but whether they are in osteosarcoma, that's not really being shown just yet. I'm interested particularly in EBV-specific T-cells, and I'm going to call them from now on EBV-STs. And this is because our population that gets affected with osteosarcoma, generally the young adults and adolescents, have a high prevalence of EBV. So presumably they will have EBV-STs in their body. And as I mentioned, but we don't know, or it hasn't been published yet, whether these cells are present at the osteosarcoma site. So what are virus-specific T-cells? So when we get sick with a virus, our body develops this immune response with our T-cells. And so the way that these T-cells work is that they recognize these danger signals in the infected cells, and these are viral antigens. So the T-cells recognize those signals, and they promote an immune attack, and eventually kill that infected cell. So what if I can make the cancer cell look like it's infected with a virus, and then have those virus-specific T-cells in our body recognize and kill that cancer? So that's kind of like the general idea of our project. So I hypothesized that there are indeed EBV-specific T-cells in osteosarcoma, and that I can deliver EBV antigens along with other immune stimulation tools at the tumor site to redirect those virus-specific T-cells to target osteosarcoma. So basically, those T-cells are there. We want to get them activated to kill the tumor. So how am I going to deliver those viral antigens? So I'm going to be using this binary CADVEC platform. This was developed by our collaborators at the Suzuki laboratory. And I'm not going to go into all the details, but basically this is composed of two viruses. One is called an oncolytic virus, and one's called a helper-dependent. The novelty of this is that helper-dependent has space and cargo capacity, so you can incorporate transgenes of interest. So this is a clinical CADVEC. Actually, it's currently being used for adults, and I'll share a little bit about the results for the adults. But it's a great therapy because since it's already in the clinic, we can potentially translate to our pediatric patients a little bit sooner. So the transgenes or the immune stimulation that this clinical CAT-VEC incorporates are IL-12, which promotes more immune T cells in the tumor microenvironment, anti-PD-L1, which is an antibody that basically blocks a stop signal at the tumor site so that the T cells can actually get activated. And so for my project, I want to incorporate in that cargo capacity the EBV proteins so that the EBV-specific T cells can recognize and kill the tumor. And finally, the other component of the CAT-VEC is the oncolytic virus, which directly actually promotes tumor killing. So as I mentioned, this binary CAT-VEC is currently being used in adult clinical trials where it has shown safety and efficacy. This is an example of a head and neck squamous cell carcinoma patient that got this CAT-VEC. Again, this is a tool that is promoting immune stimulation. And this is the scans of the patient after 30 weeks. So we are getting sustained responses. And as I mentioned, for my project, I will be using this tool that is already out there to deliver also the EBV antigens. So my hypothesis is that we do have EBV-specific T cells in the tumor, and I'll be delivering the EBV antigens to promote activation of those cells. But the first question is, like, are there really EBV-specific T cells in osteosarcoma? So that was a big part of the question. And luckily for me, through Factor, actually, and MIB agents, I met our collaborator, Dr. Livingston, and the MD Anderson Cancer Center team has expanded tumor-infiltrating lymphocyte from osteosarcoma patients. And I'm not going to go over all the data, but we have shown that actually there are EBV-specific T cells in the osteosarcoma tumor-infiltrating lymphocytes. And this is one representative patient with metastatic lung lesions. And here you can see that after I stimulate this patient tumor-infiltrating lymphocytes with EBV PEP mixes, there is an expansion of EBV-specific T cells, and that is demonstrated as release of interferon gamma through an ELI spot. And this is the representative ELI spot. And you can see that these T cells are responding to different antigens of the EBV virus. So yes, there are EBV-specific T cells at the osteosarcoma site. Next, can these T cells actually promote anti-tumor activity if we make the tumor cell look like it's been infected with that virus? So this is a proof of principle experiment. So I don't yet have the CAT-VEC that expresses the EBV antigens. So what I did is I generated tumor spheroids that are GFP labeled, and then I use this smaller recombinant virus that expresses EBV antigens only. So there's no immune stimulation here yet. 24 hours after I added my virus-specific T cells, and then I looked into tumor cell death as shown by loss of GFP. So when the tumor is healthy, it will be green, and as it dies, the GFP or green fluorescence will go off. So this is what the tumor spheroids look like when they're healthy. And then as the T cells go around the tumor spheroid, you'll see all of these gray dots. So those are the T cells trying to get rid of the tumor. So here's representative results for the cell line MG63. This is work from one of my undergraduate students, Emily. So MG63 tumor only, when you treat it with these EBV antigens, it doesn't really promote any tumor death. You see the green fluorescence going up, which is what we expected. But when you add in the co-culture EBV-specific T cells, which are my cytotoxic cells, they're actually starting to kill that tumor. And so that green spheroid starts dying, and you see this loss of green fluorescence over time, as represented by the green curve. And just to show that this effect is EBV-specific, we also did a similar experiment using the same donor, but CMV-specific T cells, so different viral antigen, and we don't see any response. Similarly, we did another cell line because we want to test this in different HLA settings. And so this cell line has a different HLA setting, and similar findings were seen where when you incorporate those EBV antigens at the tumor site, you get tumor recognition and killing by EBV-specific T cells, and you see those spheroids start to look more black and less green. And this was not seen with CMV-specific T cells. Finally, we wanted to make it a little bit more complicated, and we said, what if we add that immune stimulation with the IL-12 and the anti-PD-L1 with the CAT-VEC? So this is what a time lapse of like 5 days of a spheroid in presence of EBV-specific T cells without any kind of virus. When you adjust the EBV antigens, you can see the loss of GFP over time, but there's a little bit of residual GFP. And then when you add all this immune stimulation through the CAT-VEC, then you see that's like a stronger effect, and that tumor sphere really looks very unhealthy over time. And here's a graphic representation of how that fluorescence is being lost over time. And so our combination treatment would be the purple curve down there. And this is just our controls. And so lastly, with the spheroids, this is a really nice model because it's three-dimensional, but it's kind of hard to quantify cells. So I did a monolayer co-culture where I looked not only like tumor death, but also counts of CD8 T-cells and secretion of interferon gamma. So the combo treatment, as I call it, would be the purple bars. And you can see that the residual tumor has the stronger anti-tumor effect when you have the combination of treatments in the purple bars. And this is seen only with EBV-specific T cells. And that correlates the more anti-tumor effect with more CD8 T cells in the co-culture, as well as more interferon gamma secretion. So all in all, we did show that EBV-specific T cells are in the osteosarcoma, actually in the lung lesions, that they can be redirected to target osteosarcoma tumor cells when those osteosarcoma cells are presenting EBV antigens. And in addition, when you add the immune stimulation, you see more anti-tumor effect, more EBV-specific T cells in the co-culture, and higher interferon gamma release. So with that, this was all proof of principle. Now we're going back to the clinical CADVEC, and we're going to be modifying it to express not only the immune stimulation, but also the EBV antigens. And we're going to be testing it in orthotopic lung osteosarcoma. So right now, we're towards the end steps of this cloning to get the CADVEC so that we can test it in animals, hopefully, end of this year. And this is just a little bit of my inspiration. 2006, my brother is an osteo warrior. He's now 37 years old, and that's my prom picture, actually. And so now he's a healthy young man and father of a child. So that's a little bit of my inspiration. And of course, thank you to my mentor, Dr. Malcolm Brenner, and our collaborator, Dr. Suzuki, who developed this CADVEC technology. And of course, this work is because of Sydney. Thank you.
Before we get into questions, I neglected to mention that Ariane is also the recipient of a Young Investigator Award from MIB last year. So, okay, so now we have time for questions, and I'm going to use the moderator prerogative to ask the first one. And actually, this is to you, Ariane, and to anybody else who wants to. But so I think one of the things that parents and patients come here for is to see what's on the horizon and how soon will this be available to help patients. So do you want to give us a glimpse into the future of the viral-specific T cells? How soon do you think you're going to get them to the clinic?
Yes, is this not working?
You have to hit it.
Oh, okay. Can you hear me now? No, now I pushed, okay, now it works. Yes, no, that's a very exciting question, and it's kind of like one of the things that inspired me to pursue this work. So the advantage of this project is I'm taking advantage of a tool that is already out there, clinically available for adults, this CADVEC. So, and as I mentioned, we're just towards the end of completing the new version that has the EBV. So next step is, and towards the end of this year, I hope to complete the animal studies, which would be required to submit the new IND and eventually like translate to our patients. But my hope is that with this having been shown to be safe in adults, that we don't encounter too many hiccups and we can translate this within the next 2-3 years.
I have it's Alex Huang from Case Western. Two quick questions, one for Betsy and one for Ariane. Betsy, question is it clear to you in the STING agonism immune activation whether type 1 interferon is the final effector molecule that mediates what you're seeing, or is it some other candidates? And then for Ariane, for EBV, is it important to focus only on CD8 positive EBV specific, or is there a role for CD4 potentially epitope that may or may not contribute to what you are seeing in efficacy? Thank you.
Thanks for that question. I don't see any evidence that it's not type 1 interferon. We have looked at a cytokine expression in the tumor models that I discussed, and certainly interferon beta is increased, and a number of other sort of downstream interferon-stimulated gene-related cytokines are active in the tumor and in the plasma of these mice. We actually have pending right now an interferon, like an IFNAR1 knockout of the tumor cells themselves, really to kind of elucidate if, everything I've mentioned about monocytes coming in, about, knocking out the CD4 and maybe potentiating the effects of diabsy. I think the question still stands as to how exactly are these tumors dying so rapidly as a result of this STING agonist. And so I think evaluating whether, interferon itself is cytotoxic to the tumors versus, whether they're actually getting cleared out by the immune response itself, I think is a critical question that's outstanding.
All right. Thank you for the question. So my question was about the role of CD4 and CD8 in our project. So for the preliminary studies that I've done, I do some phenotyping of our product before doing the co-culture assays, and I've seen the strongest responses on the products that are predominantly CD8. So when they're like 60% CD8, I see the strongest responses, which kind of makes sense because these are the cytotoxic cells. But I don't discard that there's going to be, of course, a helpful component of having the CD4 as well there. I just haven't really looked into the specifics of what happens when there's like a majority of CD4 in the population.
Hey guys, great talks. My first question is for Jeff. It has to do with in your mice that essentially clear the spontaneous tumor model. Have you guys looked at profiling any of the T progenitor exhaustion populations within those mice?
So thanks for that question. We struggle a little bit to look at that population because the tumor is essentially cleared, so there's not a lot to flow on. We have looked in the spleen, but really mainly only at that memory population, so I don't know that we see any evidence of a progenitor exhausted population there.
I have a couple more questions too. So for Richa, my one question with your PC or peptide-centric CAR is, can you comment on the on-off kinetics of that CAR versus, or your PC CAR versus like a FMC63?
Sorry, can you please repeat the question?
Yeah, I'm just kind of curious if you guys have studied the on-off kinetics on your MHC peptide complex at all with your PC CAR.
So it was very interesting to see when the peptide-centric CAR T cells got activated, they release interferon gamma. And in our neuroblastoma models, we saw that the interferon gamma released by T cells enhances the HLA expression to a very high degree. And I tested that on osteosarcoma cell lines, and I saw around 6 to 7-fold increase in HLA expression post-interferon gamma treatment.
That sounds good. And then my last one's for Betsy. It's just, I have a question. During your depletion studies post-challenge, about when do you see a CD8 depletion in circulating T cells? Because I noticed you basically do your challenge and then you start depleting at day 3 and then 2 more deliveries.
So in the re-challenge context, we deplete either the 4s or the 8s before we re-challenge the animals with the tumor implantation. And then we continue twice weekly, sort of basically for a number of weeks. Does that wasn't your question though?
Yeah, no, not the re-challenge studies, but the initial tumor challenge.
Got it.
Where you're depleting like mid-day or mid-treatment.
So right. So in that case, in sort of the acute sort of model where we see this tumor regression due to the STING agonist, so the tumor is engraft, and then 24 hours before the STING agonist, we start the depletion, and then we continue the depletion for, again, a number of weeks. And the STING agonist is only dosed twice, separated by 1 week. So basically, the depletion continues throughout. There was one experiment that you may have noted, the one where we initially did the anti-CD8 depletion, where the depletion continued it for a shorter time. So there is that caveat in the interpretation of that study. But yeah, if I still didn't get to your question, maybe we can chat after, or you can clarify further. Yeah. That must have been my error in the slide then. Yeah, we definitely depleted the T cells before we gave the STING agonist in all cases. Thanks.
I absolutely loved this session, the whole thing. I loved the intro and all the talks. Just terrific. I had a question for, I'm sorry, Richa? Did I say it right? Sorry, yeah. One of the big issues that I think we see with CAR T, I'm speaking specifically in patients in solid tumors, so in that context, is that we can target tumor cells that express the target, but that we don't see kind of the complete immune response. We do see that they're targeting it, but we don't see activation. In your setting with your peptide-centric CAR, do you think that because, I wondered, okay, so first part of that question is, have you looked at antigen presentation part of the peptides that you find could be presented by antigen happening and maybe epitope spreading? And then the second is, would your CAR then therefore be better at elucidating that more complete response than a traditional CAR?
So we anticipate a CD8 T-cell response because these are HLA-1 derived peptide, and CD8 T-cell, we saw in case of neuroblastoma a very high fold enhancement in CD8 T-cells expanding in the blood at day 14. And this was similar to what people have seen in the preclinical models for leukemia and lymphoma. So that's why we were pretty enthusiastic about it. And we also saw the T-cell immunophenotype of these cells. These cells were not getting exhausted and were persisting up to like more than 100 days in mice. So we do believe that it is a very promising approach, as it's more of a peptide-centric approach as compared to targeting a specific protein on the surface, and that specific protein might be present at maybe a lower level in normal tissues, but the peptide might not be. So targeting that peptide is more of a safer, less of target toxicity.
Okay, I guess I will ask another question. So everybody, you've heard each other's talk. Can you see any future, or can you suggest how some of these therapies can be combined? For example, the STING pathway with viral-specific T cells or CD8, et cetera. So I throw this open to the whole panel. Betsy, I see you shaking your head, so.
No, I was just briefly going to add, I think, again, getting on to the soapbox of we're only as good as the models we use, I think a critical sort of avenue for, I think, potential collaboration among all of us could be to really try to be testing our immunotherapeutic approaches sort of across models in a consistent manner. And I think for sure, Jeff and I have already been kind of working together, and I think in particular now that I'm understanding sort of what he's seeing in terms of the T-cell responses in the genetically engineered mouse model, I think that's really a critical next step for our work, is to study STING agonism in that context as well. So not a combination therapy necessarily, but more like, using, employing these collaborations to really be able to test our therapies in sort of the best models we have available.
So Jeff, do you want to respond and maybe
Yeah.
how her work showing that there's a macrophage component may help you in your studies?
Totally. I think I alluded to that a little bit in the talk, which you've seen this decrease in CD206 TAMs and things that we find were very immunosuppressive. So that's an obvious combination. But I also think even if you're going to do an EBV virus-specific T-cell, it would probably benefit from remodeling that microenvironment with a STING agonist and probably immune checkpoint blockade, because we know, EBV is one of the many things that you get virally exhausted T-cells from. So I think no one of these approaches is going to be sufficient. And then the other thing we have to realize is that when this goes into patients, it is not going to go into patients that have had a normal immune system at this point. At the best case, it's going to go in on top of MAP therapy, and I think we need to be thinking actively about what does the endogenous T-cell population, what does the EBV T-cell population look like in that context when we think about how to deploy these therapies.
So I would like to add on that, with regard to Jeff's work, it will be critical for us to know the T-cell immunophenotype in the tumor microenvironment, and that can be helpful for us to armor our CAR T-cells to further extend this peptide-centric CAR T-cell approach in an appropriate way to combat the tough tumor microenvironment. And while also for Ariana's approach, the virus-specific peptides, which will be presented on the HLA, can also be targeted via peptide-centric CAR-T approach. That will be also extremely novel and beneficial in future.
Yes, and going to the point that you brought out a couple of times, because I think one of the challenges when you're talking about peptides and HLA presentation is like a lot of people think, oh, HLA is going to be downregulated, you're going to run into that trouble. But you've already mentioned how your product already upregulates interferon. Our product and the CADVIC also promotes all that immune stimulation. So it opens up opportunities for anything HLA presented that we can think of or looking into the future. Of course, with HLA, you're thinking more like there might be restrictions and you have to know the patient HLA status, but if you can find peptides that are presented like in a variety of HLAs that are pretty abundant, I think that that's really helpful. Yeah.
Any other last questions from the audience?
I had a question. Yeah, thanks. Brian Ladle from Johns Hopkins. Maybe just one comment and a question. I actually am very optimistic about combining immunotherapy with MAP, right? It's literally the only chemotherapy regimen I give where we say, oh good, you don't have to take Bactrim. Right? I mean, these patients, their lymphocyte counts are preserved all through treatment, which maybe might bode well. After ifos etoposide, right, that might be another situation as far as T cells go and things. But Betsy, maybe a question for you and others. So as you know, we've looked at STING as well, and so I'm a big fan of your work. So one thing I wonder, for instance, with systemic STING, or we'll hear Dr. Ligon's presentation here, right, kind of the nanoparticles, right, MTP, do you feel or do you see, for instance, like in your cytokine panels, like maybe kind of a reset of, are there kind of predominant immune suppressive cytokines systemically that you might see that, for instance, are those being reset a little or kind of recalibrating, that immune response? And I wonder if, again, some of these other agents might accomplish the same thing and might be a goal of this approach. But wondered if you'd seen any of that in your cytokine panels.
Yeah, that's a really great question. Admittedly, I don't have an answer. I think, when we ran those analyses, we were sort of, we had the focus of sort of what is changing in response to the STING agonist. So I think that's a fantastic idea to go back and look at that data and sort of take a look at the baseline sort of profile and try to get a window into that. So no answer yet, but that was a great question. Thank you.
Okay, so that I guess this concludes the session this afternoon. I think we'll have a break and then back in 10 minutes, I think.
3:15. 3:15.
3:15. Okay, thank you.